Hi everyone, welcome to another episode of Vetsplained. This is Pathology Edition, in which we'll be talking about one of the most important topics with respect to external as well as internal. So this is reaction to nervous tissue to injury. This for externals can come up to like 10 marks. This topic has a very big weightage in the external as well as internal. It can come up to 10 marks as a whole, explain the reaction of nervous tissue to injury, or it can come for fill-ups as well as definition. So this main topic is divided into three subtopics. We have changes in the neurons, changes in the axons, and changes in the blood vessels. So first we'll talk about why nervous tissue is very susceptible to injury. So you may ask why is it very susceptible to injury, and it is very important. So the primary reason are of two. The first reason being the spaces in the brain, for example, your cerebral parenchyma. And your meningeal spaces. So these two spaces, these are excellent for the growth of microorganisms. So they are very susceptible to injury. So this is one of the primary reasons. The second reason being the nervous tissue. Once the nervous tissue is damaged or degenerated, it cannot grow back, or the healthy neurons cannot replace it again. These two reasons are the main reason why your nervous tissue is very susceptible to injury. Now we'll talk about the changes in the neurons. So the first we'll talk about is the changes in the neurons. So changes in the cytoplasm. Under neurons, we'll talk about first the changes in the cytoplasm. So in cytoplasm, we have this tigroid substance, or also called as nacelle substance, which undergoes segmentation and dissolution. So when it undergoes segmentation, there is two possibilities. It can undergo central segmentation or peripheral segmentation. If it undergoes central segmentation, then it is called as central chromatolysis. If it undergoes peripheral segmentation, then it is called peripheral chromatolysis. So these you will see in the case of tetanus. And the peripheral chromatolysis you will see in the case of swine fever, classical swine fever. Now, what happens after dissolution? So after dissolution, vacuoles start to appear. If you know vacuoles, these are the pathognomonic lesion of scrapie or even BSc. So scrapie or BSc में ये दिखने को मिलता है. And then after the vacuoles appear, there will be a formation of inclusion bodies. Now these inclusion bodies can be further intracytoplasmic, or it can be intranuclear. So intracytoplasmic you will see in the case of rabies, and intranuclear you can see in the case of pseudorabies, or otherwise also known as Orzesky disease. Now under the changes in the neuron, we have second part after cytoplasm, that is changes in the nucleus. So nucleus, what happens to nucleus is nucleus swells and occupies eccentric position. मतलब एकदम कोने में जाके बैठ जाता है वो. So it may also show pycnosis. This is very important. Pycnosis, karyolexis and karyolysis. It can come as a fill-up as well as their definition. So it is very important. When I talk about pycnosis, it is nothing but it is the shrinkage of the nucleus. And when we talk about karyolexis, rexis meaning fragmentation. Fragmentation of the nucleus. And karyolysis, lysis meaning dissolution. Dissolution of the nucleus. And obviously under nucleus we also have nucleolus. So nucleolus may also enlarge and they may also occupy eccentric position just like our nucleus. So now we will start the next subtopic, which is changes in the axon. Previously we did the changes in the neuron. Now we come to the axon part. So under the changes in the axon, we have first our myelin sheath. Now myelin sheath meaning this one. So there are basically two changes that is happening in the myelin sheath. First being your swelling and degeneration into the simpler lipids of the myelin sheath. Second, we have is Wallerian degeneration, where there is changes taking place, changes in both axon as a whole and myelin sheath, which is called as Wallerian degeneration. Now, another subtopic under this subtopic of axon is neuroglia. So neuroglia, also known as glial cells, these are nothing but these are the supporting cells of central nervous system and peripheral nervous system. So under these neuroglial cells, we have three major cells, which is microglial cells, we have astrocytes, and we have oligodendroglial cells. So you can differentiate these three major cells on the basis of H&E staining. In H&E staining, only the nucleus of these cells are stained. So basically, under H&E staining, you will see microglial cells. They are exactly like mononuclear cells. And astrocytes, you will see that their nucleus is larger. While oligodendroglial cells, you will notice that the nucleus is smaller than that of astrocytes. Now, these two microglial cells and astrocytes, you will find them in gray matter, while the oligodendroglial cell, you will find it in white matter. Now, after that, I have taught you about those cells. Under this changes in the axon, the main important all your definitions, fill-ups will be asked from this section. So this is very, very important. So first of all, in hypoxic and toxic condition, hypoxic meaning when there is less oxygen, and toxic conditions, there will be astrocytes will get proliferated and their increase is known as gliosis. So this may come as a fill in the blanks or a tick the correct answer or even as a definition for gliosis. So and the next one we have is microglials. Microglials are basically brain histiocytes, meaning they are the macrophages of the brain. So what happens is microglials, their main function is they phagocytize hemosiderin pigment from hemorrhages, as well as they also phagocytize fat from the necrotic neurons. But what happens is these microglial cells, they do not or they cannot digest fat very well. So as a result, what happens is they become large, swollen up ho jayenge, and this is called as foam cells, glitter cells, or Ertiga cells. So sometimes they may ask you as foam cells or even glitter cells or Ertiga cells. So remembering synonyms is very, very important. Now, when neurons are about to degenerate, both microglia as well as oligodendroglial cells move in and around the 6 cells. And this congregation, meaning the collection of these cells, these glial cells, is called as satellitosis. And the cells that are involved in the formation of these congregation of glial cells, those are satellite cells. Now, after this, what happens is there is death of the neurons. So, after the death of neurons, satellite cells invade necrotic tissue, and what it does, it engulfs this necrotic tissue. So, this process is called as neuronophagia. Now we come to the last subtopic of this main topic, which is our changes in the blood vessels. Now, under this subtopic, there's a very important term called Virchow-Robin space. Now, you may ask, what is Virchow-Robin space? So, this is nothing but it is a perivascular space around the arterioles and veins, meaning the blood vessels, which is formed by the adventitia of these vessels, matlab the outermost connective layer of the vessels, and pia mater, which is one of the layer of the brain meninges. Now, these Virchow-Robin space, it normally contains CSF, but under damaged condition, what happens is there may be accumulation of reactive cells. So, reactive cells, matlab inflammatory cells, just like your lymphocytes, eosinophils, they all accumulate here, which is now called as perivascular coughing. Which is another very important term used here. So this was it for today's video. Hope you liked it, and if you did, please like and subscribe, and then stay tuned for the next video.